Hello everyone, and welcome to this overview on detergent screening for membrane proteins. Membrane proteins are difficult to handle, to keep active and stable, and therefore finding the right buffer and detergent is a critical step in membrane protein purification and downstream analysis. In a FIDA measurement, the software fits the raw data, and based on the peak width, the curve fit leads to the diffusivity, here indicated with a blue D in the first equation, that is used to calculate the hydrodynamic radius via the Stokes-Einstein equation. Note that the residence time, indicated here with TR, is the time of the top of the peak reaching the detector. TR is linearly proportional to the viscosity. Therefore, the FIDA1 directly measured viscosity, which is part of the Stokes-Einstein equation. FIDA Bio have developed a new, rapid, and highly sensitive method to determine which is the best detergent in solubilizing your GFP membrane protein in completely crude media. No purification step required. Together with the hydrodynamic radius, the user is provided with the peak area, which is a reliable and consistent parameter that is directly proportional to the fluorescent light. The more fluorescent protein is in solution, the higher will be the peak area. Here, an example of 100 nanomolar Fitch in orange and 10 nanomolar Fitch in green. Both are measured with the hydrodynamic radius of 0.53 nanometers, but one has a peak area 10 times larger, reflecting perfectly the concentration difference. Furthermore, the polydispersity index is included in each measurement. No need to run further experiment with different technologies. The polydispersity index is a very useful parameter in detergent screening, as monodispersed samples are very important for downstream analysis like cryo-EM. The molecules are considered monodispersed when the polydispersity index is lower than 0.05. GFP-fused membrane proteins solubilized in detergents can be accurately measured in 3 minutes, and the intensity of the signal is a direct and robust indication of the amount of protein in solution. The more signal, the more protein was solubilized and stabilized in that specific detergent. Simple as that. On the left, we have a measurement, and on the right, we have the overlap of 3 different detergent performance. The results are easy to interpret. Here, the UDM was the most successful detergent, followed by C12E18 and OG. Moreover, the FIDA signal contains a lot more information which is available without additional effort or experiment. For example, a reliable and quantifiable estimation of aggregates presence in terms of spikes, intensity, and counts, and the polydispersity index. In the FIDA detergent screening, compared to other standard methods such as FSEC, an unmatched low amount of precious membrane protein material is used. Only 120 nanoliters of diluted lysate is used in a triplicate measurement, with a lysate concentration between 0.02 and 0.1 milligram milliliter of total protein concentration. The remaining dead volume is completely reusable for other experiment, and the detergent equilibration buffer of the capillary is only 50 microliters for 3 replicas. With FSEC, the sample consumption is about 120 microliters for a single replica, and about 120 milliliter of detergent buffer is used to equilibrate and run the column. Here is shown a full workflow overview of the whole process, starting from the cell lysis, solubilization, and actual FIDA experiments. To prepare your GFP-fused membrane protein, you have to 1. Lyse the cells, spin down the insoluble fraction, and resuspend it in PBS. 2. Perform a breadfruit protein assay to estimate the total protein concentration, then dilute it to 2 milligram milliliter, aliquot it, and freeze it in a -80. 3. You have to solubilize the membrane protein by mixing 340 microliters of PBS buffer, 20 microliters of crude lysate, and 40 microliters of detergent stock 10%, ending up in 0.1 milligram milliliter of total protein concentration. Mix it with a pipette and place it at 4 degrees on a top-down tilting table for 2 hours for the solubilization process. Step number 4 is an ultracentrifugation at 100,000 G for 45 minutes. 5 is the actual FIDA sample preparation, where you need 2 vials for each detergent to be tested. One is the indicator, here marked in yellow, containing the protein of interest solubilized in the desired detergent, for example here 1% TDM. And the second vial is the analyte, containing just PBS buffer with the corresponding detergent. The last step is to set up the FIDA sequence and walk away. If you test 12 different detergents, in 3 hours your data will be ready. Here is shown a visual concept of the FIDA assay, where first the capillary is filled with the detergent buffer, here indicated with detergent number 1, and then 40 nanoliter of indicator marked in yellow containing the lysate in the same detergent. Detergent is injected. When the injection reaches the detector, we will get a standard FIDA signal, and when it's compared to another detergent that, for example, solubilizes less protein, we will get a lower fluorescent signal. To make sure your assay is robust and reliable, please note that the solubilized crude membranes are usually unstable, so it is important to keep them on ice during sample preparation, use only fresh thawed samples, consider adding protease inhibitors to the lysate, and keep the instrument tray 1 at 5 degrees during the running time. To keep the sample refrigerated during the running time in the FIDA1 instrument, just load a standard capillary method and change the temperature of the tray 1 in all the steps to 5 degrees, and save the method with a different file name. For example, standard capillary tray 1 5 degrees. When you will set up your sample sequence, remember to choose the new method that you just saved. To avoid wasting time and precious sample, it is useful to test 2-3 detergent with standard or coated capillary. Here we have a sequence with 3 different detergents running in standard capillaries, marked in red, followed by the coating procedure, and we rerun the exact same samples using the coated capillary marked in green. If a significant tailing effect is visible, like in the picture on the top right, then it is a good idea to coat the capillary for your screening. Please refer to our assay optimization document to decide which is the best time to take action to avoid tailing effects. When the measurements are done and you have loaded your raw data, it is a good idea to take a quick look at them to see if all is fine. And you can start fitting your data manually, one by one, but it can be a little bit tedious and long, especially if you have many samples. So you can use our auto mode, where you can select the standard fit, the data interval, single or multiple species, and then load your dataset to auto fit, like I'm doing here. I'm going to select all the replicas of all the detergents, and you now have your dataset automatically fitted as each data point turn green, and you can see the usual parameters as the size, the peak area, which is the parameter we use to rank the detergent in this assay, and you can see how it varies between detergents. This means the different detergents have solubilized different amount of protein of interest. The fit data can be exported through the bottom generate interim report, and the saved text file can be opened in Excel, where the area values can be ranked to see which detergent has done the best job. You can also load your tailograms in overlay, one replica for each detergent, to compare the peak intensities, which is an easy and intuitive way to visualize your results. The ranking of this assay was led by DDM and UDM, which almost solubilize the same amount of protein, followed by DM, Cymal6, and so on. This concludes the guideline on detergent screening for membrane proteins. I hope you find it informative. Enjoy your FIDA experiment.